Hi there. Welcome to Mushroom Hour. Today on the Mushroom Hour podcast, we're joined by Dr. Michael Perlin, professor in biology at the University of Louisville. Dr. Perlin's interest drew him to study host pathogen interactions at the University of Chicago, and his PhD work centered on the evolution of bacterial resistance to certain classes of antibiotics and antimicrobials. But as he was near to completing his degree, a friend of his stopped by his lab and introduced him to a different microbial system, a fungal pathogen that infects flowering species in the Carnation family and replaces the pollen of the flower with fungal spores. From that point on, he was hooked. Over the years, he has continued to work on bacterial resistance, but in the last 10 years, he has shifted the focus of his lab exclusively to that of fungal plant pathogens, with projects encompassing 3 different types of pathogens on different hosts. Now, Dr. Perlin has roughly 70 peer-reviewed scientific articles in reputable journals, including Nature, mBio, BMC Genomics, Genes, Genomes, Genetics, Fungal Genetics and Biology, the Journal of Bacteriology, Eukaryotic Cell, and Molecular Microbiology. As a member of a number of scientific organizations and a prolific teacher and mentor, Dr. Perlin has a far-reaching influence on his field and has directly played an influential role in the careers of many of his PhD students. I'm excited to learn more about fungal phytopathogens that end up having a massive impact on human culture. Dr. Perlin, thank you so much for joining us on the podcast.
Darren, thank you for having me. I'm excited to talk with you about the work that we do, and given the interest you've already told me about as far as mushrooms and other fungi, I hope that I can introduce some other areas that will get you excited as well.
Absolutely. That is the joy of having on so many amazing guests from so many areas of studies. You realize how vast a topic fungi is as a kingdom of life, and how many different angles there are and things to research, really. But before we dive into your research about fungal phytopathogens, I am curious, what drew you, Dr. Perlin, to explore science?
Well, I'm not sure what the critical feature was. I don't know when exactly I decided that I was interested in science. I've been fascinated by, doing puzzles when I was really young. And I actually lost one of my grandparents to cancer when I was pretty young. And I thought, well, I want to find a cure for cancer. This is a 7 or 9-year-old. And it turned out that's not what I wound up doing. I work with this group of fungi. But, I think that was part of what drove me, and sort of curiosity in the direction of natural phenomena. That's what scientists tend to explore. And how do these things work? Again, initially, the drive was for a practical outcome. I think as I've gotten older, I'm less concerned that there's a direct practical outcome from the research that I do. I guess I'm of the philosophy that basic science will provide us with insights that, whether or not I use those insights, someone else might find a way to apply those to a particularly important problem. But for me, it's the joy of understanding at the basic level, fundamental level. For me, it's mostly biology. Although I have to say that if I hadn't gone into biology, I think I would have liked physics too, just because there's so much of a connection to sort of real world consequences. If you can imagine, like doing that bank turn, with your bicycle or your car at a certain speed, there's physics related to that. There's physics to everything we do. I just wound up doing the biology instead.
A recurring theme you brought up after speaking with many academic scientists is this idea that you eventually just start to explore things just to know how they work and really break down the fundamental questions and concepts surrounding certain life forms. And, my question as a layman is always, well, how do you apply it, Mr. Scientist? And they always answer, well, I'm not sure. That may be for someone, but I just need to understand the fundamental nuts and bolts of what's going on. And then I know that will cascade into many more applications. So very, very interesting to hear you say that. And tell us a little bit about your work at the University of Chicago, too, because that's not too far off from your cure for cancer. You studied infectious disease with Dr. Stephen Lerner.
I did. And I will actually, before I answer that question, I will say that I think the model systems that I work with, if I were under the influence, for example, of writing a grant proposal to convince some granting agency to give me some money, I could probably convince you or try to convince you that the model systems I work with have an application or relevance, at least, in terms of diseases or things that would affect humans, and that they serve as good models for trying to understand those things, or at least to make predictions about what we could do as far as those other areas of concern for humans.
Right. There's a definite applied feature of your work that's pretty apparent.
Definitely. There's a spin that, again, like I say, if I were trying to get grant money, I would definitely be in there trying to convince you the connection between this and cancer, the connection between this and whatever it is. So in terms of Steve Lerner was, until very recently, an infectious disease medical doctor. He was at the University of Chicago when I joined there. I actually was set to become a lifer, I think, at the University of Chicago. I started as a college student, and I had various options when it was time to graduate, and wound up going there for graduate school, and even did a very short postdoctoral piece of work there. And, had I not been offered a job in academia outside of that, who knows, I might have stuck in Chicago even longer. But he was a great mentor. I think a lot of his approach to science and to dealing with students is one that I've been affected by, and that I tend to apply to the people I'm trying to mentor. He was very hands-off to the extent that, he would present an idea or a problem or something that we were trying to look at, and make sure that you had sort of the fundamental training in how to work with the organism or the system, and then, let you run with it. And that was something that, it could be frustrating, because when I joined the college, I was super enthusiastic and very driven. And so, I might spend hours and hours in the lab and set up these massive experiments, only to come in the next day and go, oh my God, none of that worked. Now I need to do it again. And I tell students this too, if you are not able to deal with frustration, research science is probably not your area to go into. There are other things you can do. They're, maybe less frustrating at times, but you have to be able to deal with the frustration of an experiment not working, and then think about what might have gone wrong, or think about different ways to apply it. And also maybe not set up such a massive experiment that if it fails, you're going to be devastated the next day. You really do need to be thinking carefully about how you design experiments and taking the time to appropriately do them. So part of his approach, I think, allowed me to learn some of that for myself, to realize that you really do need to think about how you do things. On the other hand, he was also somebody that, if you're having a conversation with him, he would say, so why do you think that? And then I'd really have to think about why I just said what I just said, and we would have that kind of back and forth. I guess people traditionally think of that as a kind of a Socratic approach to learning.
I was about to say that, yeah.
And I really sort of incorporated that into my being. And it's also part of the way that I teach when I teach classes, and also in the laboratory. We'll start doing something and I'll say, so why do you think we're doing that? And people think about it. They don't necessarily get the right answer, but I think the main thing is to get people thinking, and that's always a good start to whatever direction that we're trying to go.
Absolutely. Developing those critical thinking capacities is going to help people no matter what. And you were doing some interesting work, as I mentioned in the intro, with antibacterial and antimicrobial resistance, which is something that I think is kind of a huge topic in the era of viruses and disease that we're kind of in now. That's a hugely important topic. I guess it could be many podcasts on its own, I'm sure. But what was the general overview of some of the work you were doing, and what were maybe some of any outcomes or findings that you had from that work?
Okay, so I would step back a second and say that when we're talking about host pathogen interaction, I think Steve Lerner took a really evolutionary perspective, even though we're very interested in mechanism and sort of the nuts and bolts of the molecules involved. His approach was to say, well, evolution doesn't typically start from scratch. So if you have a particular protein or enzyme that does a particular function, that's a good starting off point to make changes that might go off in a different direction, or at least might expand the function of that protein or enzyme. In other words, through mutations in an existing protein or existing gene, you might see evolution progress in a direction rather than trying to start from the very beginning and come up with a completely new way to approach a problem. So if you're a bacterial cell, they swim in some pretty rough environments, right? Like inside humans, for example.
Right.
And humans can be really mean to bacteria, right? They can provide antibiotics, which are basically designed to kill them. So if bacteria develop a mechanism of resistance to one of these drugs, one of these antibiotics, and then say pharmaceutical chemists or people that are looking into drug design, they say, well, how about if we slightly change this drug to combat the way in which the bacteria became resistant to the first drug? We're going to modify that drug and see then if that stops the bacteria. And the bacteria say, oh yeah, that hurts me, but wait a minute, I got this mechanism that I had to the first drug, the parent drug. What if I slightly, by mutation, make a change that now expands my ability to become resistant to the new drug as well? And I'm saying that as if bacteria is sitting down thinking and having conversations with each other going, yeah, what can we do to fix these guys? They're making it harder for us. What can we do? It's more that if they have that reservoir of resistance to one kind of drug or similar drugs, that's a good starting point for evolution to act on and select for mutants that have modified that mechanism so that that mechanism can now apply to new drugs, even semi-synthetic drugs that humans are designing.
How does that viewpoint then change the way we approach dealing with microbes and bacteria, this kind of more evolutionary perspective on things?
Well, I think it's a good question. And so Steve Lerner and a number of other investigators that took a scientific and an evolutionary approach to this problem of antibiotic resistance, they really saw that as a really strong lesson in terms of the way that we use antibiotics in particular, because the antibiotic era is not that long. You're talking about, I think, penicillin in the 1940s, 1930s maybe. The first aminoglycoside antibiotic, streptomycin, I think was in 1945. So it's a very short period of time that we've used antibiotics to try and combat bacterial infections. Now, that doesn't mean that bacteria haven't seen antibiotics in nature before, because a lot of these antibiotics are natural compounds produced by their competitors, right?
Yeah.
So the bacteria are seeing things that their competitive microbes are producing to kind of reduce their numbers and make the producers more competitive. So what Steve Lerner and Stuart Levy, there's a pair of twins, both of them involved in infectious disease, but Stuart Levy was involved in antibiotic resistance. And the primary idea was, if you find a good drug, don't waste it. Here's an example. If you go to the doctor with a sore throat, chances are you may have the experience that the doctor gives you an antibiotic to deal with the sore throat. True?
Right.
Yeah. Now, the thing is that as an adult, it's very unlikely that you have a sore throat caused by bacterial infection. It's more likely that you're going to have a sore throat caused by some sort of a viral infection, something related to cold or some kind of virus like that. And those kinds of infections can't be treated by antibiotics like penicillin or aminoglycosides because the virus doesn't respond to those. There's no way that those drugs could kill the virus.
Right.
So what happens is, if you take a drug that's good against bacteria and you waste it by basically flooding the environment with it, that creates an environment that selects for bacteria that are inherently more resistant to those kinds of drugs. And so both Steve Lerner and Stuart Levy were in a group of physician scientists that said, no, you need to withhold these drugs. You need to use them for those cases where they're absolutely necessary to kill an infection, to cure an infection that otherwise might cause the patient to succumb or at least might make it harder to cure the infection. So there's a direct application. So think about evolution is going to act on these bacteria, and we can't assume that we can just take this stuff like water. You can't be spreading that in the human environment and also the agricultural environment, because it turns out that a lot of farmers use antibiotics to try and protect their animals that they're raising in agriculture. But guess what? Those farmers are not trained in infectious disease, and they're not trained in what kinds of dosages they should be using, and whether or not they should be even using a particular drug in this context. So you have instances where you have an increased prevalence of salmonella, for example, in the dairy industry, people dealing with chickens and also cows and such for producing dairy. They're giving their animals these antibiotics, and the result is the sensitive bacteria get killed off. The resistant ones are the ones to stay. And I tell you something, we don't want drug-resistant salmonella out there.
No, I mean, the over-application, this speaks directly as a caution against that over-application of antibiotics. And I think all of us are becoming a little bit more aware of that. But I think you just explained it beautifully. And it does speak to the fact that having this evolutionary perspective that, everything is an evolutionary environment for bacteria, and they're constantly responding to inputs. So let's not waste antibiotics and make that part of their evolutionary selective process to suddenly we have these mutant bacteria that can resist everything we're putting out there.
The other thing is, if you do that, it makes drugs that would have been good less useful, and in some cases worthless. And we do not want to go back to the pre-antibiotic era as humans. I don't know how many humans have been saved from infectious disease by the use of antibiotics. It'll be a challenge as we go forward because of this increased resistance in bacteria to find drugs that are effective in treating infections.
So a more targeted application to these already very effective drugs that we've developed. Yeah, let's not try to go back and start from scratch. I mean, we kind of have a limited arsenal of what we can pull from nature to make antibiotics. So let's not try to start over. Well, I mean, that's a huge, fascinating, hyper-relevant world that you were in. What then pulled you into studying fungi in your studies of host pathogen interactions?
Well, I had a buddy, we had done some professional and some other work together, and he and I were both finishing up our PhDs. And he came by the lab one day and he said, hey, let me tell you about this system I'm working on. And actually, the way he phrased it was, have you got any DNA I can put into these guys? And I said, well, yeah, we had all sorts of DNA. We work with various kinds of DNA. He asked me if I had any DNA lying around that I could use to put into his favorite organism. And so he told me about his favorite organism. He was finishing his PhD with two real father figures in the area of the kind of organism he was working with. These are really prominent fungal geneticists. Yeah, a little bit ecology, but very much fungal genetics. And he told me about this organism called Microbial violaceum. Actually, at the time, so things change. And you were talking about, at least before we started recording, you were talking about taxonomy. At the time, this organism had been called Eukaryotic Cell. And it was thought that it was related to a number of other species of fungi called Eukaryotic. And it's been sort of known of probably for about maybe 300 years, this fungus, although not completely understood. In fact, well, obviously it's not completely understood, or I wouldn't be telling you about it now. I'd be working on something else. But not well understood at the real fundamental level. Understood in terms of, you could observe it. So if a lot of your listeners are the kind that like to go out into the forest and look for mushrooms, for example, there's observational kinds of things that have been done with it for years and years and years. In fact, the one I'm going to tell you about, so it was called Eukaryotic violacea, and now it's actually called Microbial violaceum. So this is a fungus you mentioned in the introduction that infects flowers in the Carnation family. Now, that's not the only group of plants that are affected by this fungus, but it's the main big group. And so it typically infects wildflower species in this group of flowers. And what it does is it replaces the pollen in the anthers of the flower. So that's the male parts with the fungal spores. And Linnaeus, do you know who Linnaeus is?
The godfather of taxonomy and binomial nomenclature, yeah.
Exactly. And if your listeners didn't know that, this is the guy that developed the whole, genus species way of naming organisms. So it turned out that he had mistakenly named a species of flower a different species than this other species because they looked different morphologically. And the big difference was if you looked at the anthers, they were a different color. And it turned out that the new species he named was an infected member of the original species.
Okay, that's incredible. That image of the anther having its pollen replaced by spores, just incredible to see how fungal pathogens affects plants like that. And clearly it makes a pretty big difference if Linnaeus classified it as an entirely different organism.
Somebody later on, we don't need to go into that, but somebody later on pointed out to him, no, I think you maybe miscalculated on this one. This is actually an infected, at the time they didn't really know exactly what it was, but this is just an infected version of the other species that you've already named. And in fact, one of my colleagues, Michael Hood, who's at Amherst College in Massachusetts, has gone into herbarium specimens that are, 50, 100 years old and looked at some of the ones in there and found, in fact, the spores that are in these infected flowers, whether they were known to be infected or not, and has been able to at least isolate DNA from them and figure out what are these particular isolates of the fungus? How are they related to other isolates of the fungus that we have currently?
And that gets into this huge question of host specificity when it comes to fungal phytopathogens. But I guess the Microbotrium violecium, that is a smut fungus, correct?
It is.
And so that seems to be kind of the central focus of your work now, is specific varieties of smut fungi. Kind of where do they fit into this broad spectrum of all plant infecting fungal pathogens?
So there's lots of different plant pathogens out there, and I may be anticipating some of your later questions, but emerging disease caused by fungal plant pathogens is probably the single greatest threat we have. That and climate change, of course, single greatest threat we have to human agriculture as far as plants.
Wow.
Yeah, I mean, I can give you a couple examples. Maybe you've heard of Fusarium banana wilt. Is a disease of bananas that is threatening the banana that we typically have, in the store, in the grocery store. That's not to say that there aren't banana alternatives, but they may not ship so well, and they're not what the consumer is used to.
Bananas are under threat. Okay, now I am concerned.
Oh yeah, you should be. Although there are scientists who are also concerned about it. I mean, the Cavendish banana is sort of the common banana that we eat. And these pathogens that are going after banana threaten to basically eliminate that as an option for us. And so the ways that people are approaching that include trying to use traditional inbreeding methods or outbreeding methods to select for resistance to those kinds of infections. And that's being sort of in parallel coupled with sort of, and I don't know if your listeners will appreciate or be afraid of this, but more GMO approaches. So basically not traditional breeding for resistance, but going in and actually either adding genes or taking out genes or modifying genes in the banana so that it won't be able to be destroyed by these fungi.
That's something I've spoken with one other guest about fungal pathogens just in the context of peanuts. But I guess this is an aspergillus, more of a mold fungi, and the concern that most organic peanuts are likely have this aspergillus somewhere in the actual output product. It's a super deadly pathogen. But I think these are great examples of the fact that fungal pathogens are one of the most impactful things on humans, even though a lot of folks like myself who are amateur into mycology and into foraging, we don't think of fungal pathogens first thing, but the impact is absolutely huge. And that's why it's such an important thing to be aware of and to understand how these organisms work.
Definitely. Yeah, so these are important organisms. And in terms of the microbotrium and host specificity, I think that's the thing that really fascinated me about them. Because if you look at what we called, back in the day, we called it Eukaryotic Violacea, and now Microbotrium violaceum. When I started being interested in this fungus, what we thought was, here you had a species of fungus that infects maybe hundreds of different host plant species.
Oh, wow.
And yet, if you isolate the fungus from a particular host species, those isolates are host limited. They can only infect that species, or at least that species and a few related host species. And so I thought, well, what's the basis for that? If I start out from a perspective of trying to understand mechanism for how organisms do what they do, how does that work? I since was involved in discovering that the reason that you have this huge range of hosts that this fungus can infect is that what we call Microbotrium violaceum is not a single species of fungus. It's a complex of fungi. And what brings them together, what makes them the same, is sort of the way in which they do infection. And then after that, what distinguishes them is which host species they're able to infect.
Okay, so host specificity is a differentiator here. What can you tell us about the basics of the life cycle, the physiology, maybe the reproductive strategies of these different Microbotrium species, and do they share those in common?
If we talk about the life cycles for these guys, they go through the same processes. So these fungi are able to mate. So they have multiple different morphological stages throughout their life cycle. But the simplest way to break this up would be they have a yeast-like form, so single-celled form of different flavors, if you will. So they're equivalent of the genders or sexes that we have. In order for them to reproduce, they have to find a cell of the opposite mating type or gender in order to continue the process. And so the reason I brought that up is because you can take individuals of different species of Microbotrium and they will initiate that mating process. So they have what are called pheromones. It's kind of analogous to the pheromones that other animals have. Yeah. And one cell will send out a pheromone, and the opposite mating type cell actually has a surface cell receptor that recognizes that pheromone and not pheromones from completely unrelated fungi. And as a response, it will change the way it engages in gene expression, and the two of those guys will get together and they will fuse. If they're compatible, they'll fuse. And then instead of being yeast-like, so for your listeners, yeast-like cells tend to be sort of egg-shaped, or for one of the fungi I'll talk about, sort of cigar-shaped. And they have one copy of all of their chromosomes. Humans, for example, we have two copies of all of our chromosomes. And the only time that that's not the case is when we produce reproductive cells like sperm and egg.
Is this haploid and diploid?
Yes.
Reaching back into my biology, okay.
Yeah, exactly. And haploid cells have one copy of each chromosome. Diploid cells have two copies of each chromosome.
Right.
And those two copies may not be identical, but they're going to be very similar and at least encode information for the same kinds of traits. Mm-hmm. So like if you have a chromosome that codes for, this is a real oversimplification, but if you had a chromosome that codes for eye color, the other chromosome that you have, the copy of that, will also code for eye color, but it may not be the same eye color information. One might be for blue eyes, the other one might be for brown eyes. And together in the diploid cell, you have both sets of information, and how that plays out depends on the particular trait.
Yeah. And so they're mating as these yeast-like structures. And you're saying, even in the example of this Microbotrium, if they are differentiated species, that they associate with different hosts, they'll still at least engage in that stage of reproduction.
Right. That's exactly right. And that's what makes it even more complicated, because if they're able to do that, the next stage actually is to infect the plants. So when they mate, these haploid cells will change from yeast-like cells. And yeast-like cells depends on the type of yeast, but basically they reproduce by budding. So a little bud pops off of the cell and it grows and grows and grows. And eventually the mother cell lets go of it. And then you have two cells. And that changes if cells are effectively mating, because what will happen then is they will fuse, and then they will produce what's called a filament, kind of a long extended structure. And it turns out that for plant pathogens in particular, that filamentous structure is typically what they use to penetrate a host.
Right. And that did seem to be kind of the central theme in your work and reading through it, is this critical transitory point where budding reproduction structure turns into filamentous structure. And suddenly that's when it becomes, an actual danger, an actual causes disease in the host. But just so I'm understanding, so in this budding reproductive form, technically the organism's still expanding. And then we can get into, I guess, then what causes it to, is it just the act of reproduction that causes it to form a filamentous structure? It sounds like there was some other environmental influences going on as well that dictated whether or not it would become filamentous or just remain as this less threatening budding yeast structure.
That's a very good point. So as I continue to talk about the systems that we work with, one of the sort of areas that we tended to focus on with one of our fungi are the kinds of signals that are required for these cells to switch from a basically benign growth form in terms of plant pathogenesis into one that is infectious and that can cause disease.
Right.
And one of the things that we've noticed is that typically cells that are starved for nutrients are ones that are primed to mate. And this is actually an area of research that we found some very interesting connections there. But when they are starved, they have two options we found, these haploid cells. They can either filament the way that some model yeast cells do as a response just to starvation, or if there are potential mates out there, instead of doing that, they can say, well, it's time to reproduce. I'm starving for these nutrients. I should find a partner. And it turns out that mating does depend in many of these fungi that make this switch. Mating requires starvation, whether it's starvation for nitrogen, which is a very important nutrient, or it may be a combination of nitrogen with other kinds of nutrients as well.
A tough concept to grapple with because the organism's still expanding even without that filamentous reproduction trigger that causes it to engage in that stage. But that just kept coming through as really the central point. Now, I should back up though and ask, what questions was your lab looking at? What big questions were you trying to answer? Maybe that was one of them. And then what fungal species were you working with? Because we know you started with the Microbotrium violaceum, but it seems like you've moved into a couple other of those of Smut type fungi as well.
So first of all, when I started working with Microbotrium, one of my primary interests was in trying to understand host specificity. Right. And that was a real challenge for me because, first of all, I was not trained formally as a person to work with fungi, and I was also not trained formally as a person to work with plant species. So I got sucked in. I was seduced by the system. And actually, I'll tell you a little bit more about that system that really got me interested.
Yeah.
And there were certain things about that system that were lacking compared to other systems, and that actually kind of slowed us down for a little bit. So yeah, my initial interest was in host specificity. And I gave you kind of a background that we initially thought we were dealing with one species of fungus that infect lots of hosts with very exquisite host specificity. And I later found out, and additional collaborators sort of pursued that even more to realize that we're dealing with lots of species. So this life cycle that I told you about, you've got these mated cells of opposite mating type come together, they penetrate the host tissue, and then the main place where the genetic information is stored in the cell, the nucleus for those guys, those two nuclei, they actually remain independent in this filamentous structure. So they don't get together. They're still there. They're in a fused cell structure, but the nuclei remain separate from each other. And there are a lot of people investigating this too, but there seems to be some kind of communication in terms of gene regulation between the two separate nuclei in the same cell that affects the correct development of the fungus further on. And so what you have then is this filamentous structure that penetrates the host tissue. You get further development. Either you expand the amount of that filamentous source of biomass inside the plant, but then eventually you get to the structure, which is where the spores are going to be produced. In the case of these anther smut fungi, the ones we're talking about, Microbotrium, that happens in the flower. In the place where the flower is going to develop, you get those two nuclei will then fuse when they're in the right phase. And then that will produce these spores that are then diploid, just like our cells are diploid. And then the flower emerges and you have, instead of the pollen, you have those spores. Now, if you think about it, when a pollinator species visits that infected flower, it's looking for pollen. It probably isn't going to find much, and it's going to carry with it spores of the fungus to the next naive flower. So if you think about this system, it's an example of a sexually transmitted disease.
Wow.
Right?
So you were essentially studying a sexually transmitted disease, but is that the primary benefit it derives from its host? Is that ability to reproduce more effectively? Is it also drawing nutrients? I guess, what is that pathogen really getting out of using that plant?
So I would say, first and foremost, if you strip away everything else, all the other trappings, all organisms are trying to reproduce.
Right.
At the fundamental level, that's what they're trying to do. Now, you asked a very good question about inside the plant, though, because they are deriving nutrients from the plant. And these particular types of fungi, smut fungi, are part of a group of fungi that are called biotrophic pathogens. And what I mean by that is they have to have living tissue in order to reproduce. Okay. There's a completely other group of pathogens called necrotrophic pathogens, and they intentionally damage host tissue, break it up, and then they suck up the nutrients from the diseased tissue. Whereas these guys, they don't really want to kill their host. That's not really their goal. Their goal is to derive nutrients from the host and then get to a point where their reproductive products, the spores, can then be released and start the cycle over again.
And the biotrophic pathogens, then, does their host necessarily die from their infection?
No.
Or can it live? Okay.
It can live. And in the case of Mycobotrium, they do have a fitness cost for their host. Aside from the, taking nutrients, they're actually castrating their host. So they're basically eliminating the ability for the male parts to participate in reproduction. And basically they make those flowers sterile, which does have a fitness cost for their host species. Their hosts can reproduce vegetatively, a lot of them. They send out runners and send up new plants. But in terms of the genetic diversity that we associate with sexual reproduction, that's going to be minimized for the infected flowers.
Wow.
Now, there is one other example I want to bring up. This is also why I was seduced to work with these organisms, and why some of my students were seduced to work with them as well. If you have dioecious species of host, and dioecious means that there are male plants and female plants that are separate. Okay. So you'll have male plants that have the anthers and female plants that have the ovaries and those parts. If Mycobotrium infects a female plant of one of these species, that should be a dead end, right? Because they don't have the anthers, there's no pollen, what's going to happen? So what does happen is the fungus will cause the ovary in those female flowers to abort, and pseudoanthers will be produced in those flowers. So you're essentially creating pseudo-male flowers. And in those pseudo-male flowers, the fungus will produce its spores and continue its life cycle.
That's absolutely mind-bending. And I had that question of how do these pathogens that you've studied end up changing the plant's physiology? And it doesn't get much more drastic than that.
Yeah, I mean, it's just like your parents told you about staying away from smut, right? You got STDs, you got, sex change. It's, all those risks are associated. On the other hand, it's fascinating, right? Why would we not want to study those kinds of things?
It's hard to stay away. So we kind of, before we got to that big picture about the Mycobotrium that you just went through, we were getting into that idea of this trigger point for creating this filamentous formation. Was that really the question that your lab was looking at, whether it be with this fungi or any of the other model fungi you were working with? What were some of the big questions you were trying to answer? Was it just understanding the life cycle you just laid out more in depth, or was there any specific research question you were going after at different points in time?
To restate, initially I was interested in host specificity, and I haven't put that one aside. I'm very interested in that. And fortunately, we've, in the last 10 years or so, we've started to develop the tools to be able to address those questions. Myself and collaborators, I have some really good collaborators, and we're really starting to zone in on what we think may be important in terms of host specificity. But those were my initial questions. I would just by saying that when I started working with these fungi, I thought, oh man, this is such a cool system. We're just going to make progress. This is, I guess, to date myself, this is over 30 years ago, right? It's about the same time that I started work at the University of Louisville. So when I joined the University of Louisville, not long after I joined, we won the NCAA championship in basketball. We went to the Final Four. We were the champs in basketball. I thought, wow, this is cool. I'm making this move to this university. They're really basketball powerhouses. And then I got this new system that I'm going to be working with. This is just great. It turns out that it was about 30 years later that two things happened. One, that was the next time the University of Louisville won the basketball championship. And two, that's about the same time that we started to develop these molecular tools to be able to really functionally study these fungi, the microbiota.
Right.
So there was a lot of frustration along the way. You got frustrated students that they need to graduate. They need to have a dissertation topic that will allow them to come up with a body of work that's worthy of earning a PhD. And so along the way, we discovered is not the right word. It's kind of like this idea of Columbus. Some people just celebrated Columbus Day for discovering, the new world. I mean,
right.
It's not like there weren't people there already, right?
Exactly.
So in this case, I didn't discover this fungus. I became aware of other smut fungi. The one that I started with probably about 20 years ago, or a little bit more than 20 years ago, is one called Eukaryotic, which is a smut that infects corn or maize. And it turned out that about the time that I started working with that organism, there were a lot of molecular genetic tools that had already been developed for that fungus. And so what we were able to do is study some of the things we were interested in with Microbacterium, initially by using Eukaryotic as a sort of an alternative organism to test our hypotheses with about the things that we were studying in Microbacterium. And then along the way, it turned out that there were things that were interesting in Eukaryotic in and of itself that have also propelled us over the years to study that organism.
So being another fungal smut pathogen, it's shared much of that similar life cycle, that key dimorphic ability from turning from a yeast cell into a filamentous cell. That was all the same even as you started studying the Metis organism.
That's true. I will point out one other thing about Metis. So I think we talked about this, but if I were under the influence of writing a grant proposal explaining why my work is important for human concerns, I could easily make a case for studying Eukaryotic Metis as a model for understanding cancer biology. For example, it turns out that Eukaryotic Metis, instead of replacing the pollen in anthers, actually causes the plant to produce tumors or galls. And it's inside those tumors that the fungus develops and eventually produces its spores that then the tumor ruptures open, and that's how the spores get out.
Wow, that's vivid. So there's no way that could be extrapolated to human cancer, could it?
Well, it depends. Are you thinking you might want to give me some research money? I'm sure I can make a case for it.
You can make the argument for us here.
I mean, for example, if you talk about model organisms, a good model organism is one that is relatively easy to study and that you can draw connections between that and what we consider to be the important organisms that we're really trying to study. For example, Saccharomyces cerevisiae is the yeast that's used for brewing and baking. And that's a powerhouse in terms of molecular genetics of a single-celled organism. It's also a fungus. But it turns out that it's not a perfect model for humans and human disease because it's missing certain things that humans have. For example, you're familiar with the BRCA genes? These are the genes involved in breast cancer.
Not too familiar.
Okay, well, so Angelina Jolie several years ago made an announcement. She'd had a genetic test. She had a mutation in a gene, I think hers is BRCA2, that made it highly likely that she would, during her lifetime, get breast cancer, ovarian cancer, or both. And so she took pretty extreme measures, but she wanted to make sure she wasn't going to get that because you don't want to get that. And the treatment is really harsh and not everybody survives.
Right.
So it turns out that Eukaryotic has a gene that's related to that particular gene. It's a gene that's involved in DNA repair. And so part of the reasons that it's associated with cancer, like breast cancer, is that if something happens to that gene, then the cells can't repair DNA damage, and that leads to some of these changes leading to cancer, if that makes sense.
Yeah, it does. So maybe the vector of changing that gene and not being able to regulate DNA appropriately is what's actually leading to cancer taking over.
Exactly. So Usalago has a gene that's related to that. And so you could use Usalago as a model to study how does that gene normally work and,
Right. And what disrupts it.
Exactly. Yeast, so Saccharomyces, they don't have a gene like that. So it points to the limitations of some of the models that we use. And it also argues for sort of expanding our model systems that we use for studying various kinds of disease, for example.
Yeah, now it sounds like the Metis ended up being sort of the model organism you transitioned to better study the microbiome. Is that correct?
That is correct.
And I guess why is that? Was it just easier to work with? What was it about Metis that made it a good model?
Well, let's think about if you are a mechanic or maybe a carpenter, you're a tradesperson, you generally have a toolbox that you bring with you.
Sure.
And the better stocked your toolbox, you're better able to handle different kinds of tasks that you're set to. The toolbox, the molecular genetic toolbox of Usalago is quite extensive. Even when I started working with it, there were lots of different ways you could genetically manipulate that fungus. You could tag genes so that when they were expressed, maybe the organism might glow, have a fluorescence, and that would give you a measure of how extensively that gene was being expressed, but also it might allow you to determine where the protein that is being tagged normally is found in the cell. And so there are all those kinds of tools that were available when I started working with Usalago. Now, over the course of the next, I'd say about 8 years, the entire genome sequence, so that basically all of the genetic information, we determined the nucleotide sequence for that. And in that, that really opens up a wide range of things that you can learn about an organism once you can see all the predicted genes. Now, having said that, there are a lot of genes, even when you see them, you look at them and you go, I still don't know what that does. But the way that geneticists tend to do things is we say, okay, we got a particular gene. Let's see what happens if we make a mutation in it.
Right.
How does that change the behavior of the organism? And that's sort of our modus operandi for studying these organisms is make a mutant and see how it's different than the wild type. And you could do that with Usalago. You could do that a lot earlier than you could do anything like that with Microbiome. So as I say, I started out with Microbiome. There was a transition period where most of the people in my lab were studying Usalago because it was a good way to get graduation happening. If you're a student,
Right.
That's always an important thing, right?
Right.
And then what happened is about 10 years ago, about 10 years ago, we got a grant from the National Science Foundation to determine the genome sequence for Microbiome, for one of the species of Microbiome. And in the course of doing that, we learned so much about the organism, at least predicted to know about that organism, that it allowed us to develop additional molecular tools. And it really has driven a major area of my lab research, I'd say for the past, once we got the sequence, for about the past 5 or more years, it's really made it so that there's a large working group in my lab that focuses on that organism, on Microbiome again. We haven't stopped working with Usalago, and actually we've even picked up another species of smut fungus that infects corn plants that's called Sporozoarium. But having all those options, all those models in the lab really it's intriguing because it allows us to start making connections between those very different fungi. I mean, right. Usalago and Sporozoarium are relatively similar, although they have their own special differences, but they're both very different from Microbiome. And so it's kind of interesting to be able to make connections between and across those different fungal systems.
Absolutely. And that was one of my big questions was, can your lab's findings on these different fungal pathogens then be extrapolated to other, at least, smut pathogens out there to give us an idea maybe of the mechanisms, or are we going to have to do this rigorous process with each and every organism that affects different plants? And it sounds like you'll at least have a better starting point for understanding these organisms based on the research you've already done.
I would agree. And I would say that I would put up a caution that, just because something works in one fungus and we've been able to tease apart the different mechanisms relatively easy, doesn't mean it's necessarily going to work as well or that it's going to work the same. In many cases, it's not going to work the same in another organism, even if it's a smut fungus. But even so, what happens is you don't have to start from scratch typically.
Right.
If people have figured out ways to approach these different pathogens, you can apply them in many cases, apply that knowledge. That's what's great about science, to try to understand other organisms that are less well studied.
It sounds like then you started with that question of plant host specificity. You started exploring other organisms. Inevitably, you start going down different pathways, trying to answer different questions. Were there any other main focal points of research? For me, I would think that there would be this interest in figuring out what happens when it turns from yeast to filamentous fungi that's causing this infection. And I would think there'd be other people like giant agricultural interests who would want to figure that out as well. But was that the case? Or were you guys more interested in just identifying the actual genetics and identifying the workings? Or did something come to the fore like that, some other big question as you continued?
So to answer that, I would say that early on when we started working with Eusolago, this whole concept of switching from a yeast-like phase to a filamentous phase is, I think, what attracted me to the system in addition to the tools that were available. So Saccharomyces is able to switch from a yeast-like form to a filamentous form when it's starved for nitrogen, specifically when it's starved for ammonium, which is like one of the most accessible forms of nitrogen.
Okay.
It turns out that many fungal pathogens, require that switch either from a yeast-like form to a filamentous form, or in some cases, some human pathogens from a filamentous form into a yeast-like form in order to cause disease. And so that did, for a long period of time, we haven't given it up, but for probably 5 to 10 years, we were very focused on what it is that causes Eusolago to switch from a yeast-like form to a filamentous form. Now, there are actually at least 2 and possibly 3 different kinds of filamentous forms that Eusolago presents. One is when they mate. So we talked about that. When they mate, they produce this filamentous form that's required in order for the fungus to penetrate the host plant. In the laboratory, you can observe this on artificial media. Typically, what we do is we'll mate Eusolago cells on a media called charcoal media. And the reason we do that is if they're successful in mating, they'll produce what's called a white aerial hyphae, a fuzz. And that white fuzz is obvious against that black background. So we can, just visually, we don't have to use a microscope. We can just look at the plate and say, yep, they produced the fuzz, so that was successful mating, or no, they didn't, so they're not mating well. So that's a type of filamentous growth that they do. And it's still on the plate is slightly different than if they were in the plant. If they're in the plant, those filamentous structures would actually branch through the plant during the process of infection. And so it's even a more developed stage of filamentous growth than what you see on the plate. The plate by its nature is sort of artificial. But the third type of filamentation that Eusolago will do, even haploid cells, so the yeast-like cells, if you put them on media that is low in ammonium, they will produce filamentous-like cells. So that if you look at I don't know if you're familiar with looking at colonies, like bacterial colonies on a petri dish, they usually produce these nice discrete dots on a plate.
Right.
And each of those dots is not a single cell. It represents probably millions of cells that are essentially the same cell, except clonal, right? Well, what you'll see is a normal colony is smooth of Eusolago on a petri dish. But if they're starved for nitrogen, they produce a filamentous structure that emanates from that colony. And that's because the cells are becoming filamentous instead of yeast-like. And a way to think about this is if you're starved for nitrogen, a human, say, you're going to go out walking looking for nitrogen, right? You're not going to stay put.
Right.
But fungi, they don't have motility. They have no way to move around. So if they land on a plate, a petri dish in a certain location, they're stuck there. Unless they produce a filamentous structure that extends from where they landed, And so the human way of trying to think about what fungi would be thinking, if fungi could think, is you're stuck on this spot in the plate. There's not enough nitrogen for you to live. Let's send out structures to search for nitrogen and forage for sources of nitrogen that are away from where you initially landed on the plate. And so that became kind of an area of focus for us. And part of the reason it became an area of focus, we've talked about getting grants and such like that. Well, the Eustalago system, Eustalagomatus, is not as large as people that study yeast, but it's a lot larger than the number of people that study the molecular genetics of microbiome. And it's a very competitive field. Let's say there's maybe 50 to 100 labs in the world that study Eustalago. It can be very competitive. And so if you are trying to get into that field, either you better be super brilliant, which is not me, or you need to find you talk about ecology, you need to find your niche. You need to find your niche in this ecosystem and study something that maybe you're not going to be stepping on people's toes, but is interesting enough for you to, spend years trying to figure out what's going on. And it turned out that what we tended to be interested in was an area that had not been as explored about Eustalago as other areas that were more directly associated with pathogenicity. And so we spent a number of years trying to study the molecules involved in how the yeast-like cell, when it's starved for nitrogen, is able to sense that and then switch its growth pattern.
Yeah, I mean, that's incredible. So this is that third stage of filamentous structure that you're talking about. And just so we have a visual, I mean, is it one cell just changing its cell wall to emit this structure? Is it multiple cells coming together to make a structure? What does it look like?
It's probably a combination of both. It turns out that it's hard to see that if they're growing in broth, so they need a surface on which to extend out. So that's why a petri dish is a perfect place to see this phenomenon. And it turns out that, what you're seeing is individual cells are elongating and becoming different in the way they extend. Normally, if you take a yeast-like cell, it can bud from either pole of the cell. It doesn't have to be from one direction. But when they're starved for nitrogen, their direction becomes only in one direction. So it's like from the cell, they extend out and look for, again, this idea that they're looking for nitrogen. It's directional from a particular cell. Now, in a colony, you've got multiple cells. So if you look at different ends of the colony, you have different cells that have extended out in different directions. But it's basically changing the morphology of the cell from this sort of cigar-shaped, in the case of Eustalago, to one that is a lot more filamentous or thread-like, extending out from the initial source.
Yeah, I mean, that's a wild change to have happen in a cell. And I guess, does that also imply pathogenicity? I mean, that third form, does that also then, have the same disease effect on the host?
That's a great question. The answer is no, absolutely not. So those haploid cells, they're doing their thing because they're starved.
Right.
And our most recent work that we're getting ready to publish now is related to why do haploid cells do that? And again, the idea is that we think that in terms of their survival, it's a way for them to forage for nutrients in the place where they get stuck.
Right.
And we think that particular trait predated their ability to mate and essentially have sex with opposite cells.
That was my question, is why not reproduce if you're in search of food? That seems to be the default and the first microbiome.
Right.
So they have some even older adaptation to just be a single cell and kind of fend for yourself.
Right. And then as sex develops, so as the possibility for sexual reproduction developed, what we think we found is that there's a connection between the parts of the organism's genetics that controls the ability to mate and the sensing of nitrogen or ammonium availability. So that basically these cells are given a choice. If you're a haploid cell and you're starving for an essential nutrient like nitrogen, you have two choices. You can either filament to go look for it, or you can say, is there a partner out there that we can get together with and now undergo sexual reproduction? That's why we think that this connection has developed between the mating program and the nutrient sensing program so that a haploid cell has a choice given the conditions of its environment.
And now, is it similar proteins or genes involved in making that decision whether to reproduce or, just a haploid form that structure? Or is that one of the questions that you're looking at?
That's what we're still trying to tease apart.
Yeah.
So we know that the simple decision, it's not a simple decision, but the decision about sensing available nitrogen has to do with the protein that's actually found in one form or another all the way from bacteria to humans. And what this protein is, it's a component of the cell membrane that basically sits there and it says, how much ammonium is available? And if the amount of ammonium is limited, then it participates in a series of what we call signaling events that tells the internal workings of the cell, you don't have very much ammonium. You better change the way you're growing.
Right.
And in general, if you want me to explain what signaling pathways are about, basically the idea is you've got a cell and you've got sort of the internal workings of that cell that decide, what genes are going to be expressed, when are they going to be expressed. But ultimately, a cell needs to respond to changes in its environment.
Stimuli.
Cells that don't do that are going to be selectively at a disadvantage. And so there are cues or signals in the environment that then need to be transduced or conveyed to the genetic system inside so that things change.
Yeah.
You change in order to respond to those environmental signals. Now, ammonium happens to be one of those. And so ammonium is a signal. I mean, it's also a nutrient, but in this case, it's both a signal and a nutrient. And the amount of it that's available is going to cause gene expression changes in the cell.
Probably because it's a nutrient, it's a signal, because the organism has learned that it needs to key into the availability of that nutrient. And obviously, for anyone that's interested, they can read more of your work. And I know that you list exactly what proteins you guys are looking at, their functions in this process. But again, looking at this applied angle, and maybe this is just, has there been any thought of, maybe not for this third form of filamentation with Metis, where it's just one haploid cell reaching out on its own, but has there been a thought of trying to affect that communication pathway in order to prevent the actual signal that would cause it to mate with another cell and form the pathogenic filamentation that happens when it mates? Is that something that's being looked at all? Is that not very practical?
I don't know if someone specifically is looking at that. I think there's a reason to suspect that could be an approach. I would put forth a caution, though, and that is that when you're dealing with fungi, you're dealing with organisms that are not that different from humans. It turns out that fungi are a lot closer to animals than they are to plants. So if you start providing chemicals that are going to interfere with a particular process in a fungus, you better be sure that that's not going to have an adverse effect on the analogous systems in humans, or if there are any, the analogous systems in plants. So you have to find something that's very precise. We talk about surgical strikes. Unfortunately, those aren't as surgical as we like them to be. But
Right.
that's what you want, right? You want something that's very focused to target that organism. And I think it may be possible in the future to develop something that basically prevents mating. So if you can prevent mating between cells of the fungus, that's sort of an initial step. I agree that could potentially be an interesting way to approach the problem.
And you just brought up this huge concept of unintended consequences that I know is another pillar of your work, which is the cautionary side. So I guess, do we want to stop fungal pathogens? Because my mind, as I'm reading your research, says, okay, they've discovered so much about how they work. Maybe now we can stop the blight of the fungal pathogens, save the corn, save the bananas, whatever it may be. But could there be these unintended consequences from doing that? Is that even a focus of your work?
Well, so I guess what I'll do is jump ahead to what we're doing right now.
Okay.
At least one of the major foci in our lab. In knowing the genome sequence of an organism, you can start making predictions about what kinds of proteins are being produced, in some cases you can learn about how genes are regulated, but you start to learn what kind of inventory they have as far as kinds of genes and what they might be used for. And it turns out that fungal pathogens in particular, but there are bacterial pathogens that do this as well. Oftentimes they produce proteins or compounds that are used in manipulating their host. So, for example, I have a colleague that works with salmonella, and salmonella actually produces proteins when it infects human cells, not just the gut, but when it gets into human cells. It will use these proteins called effectors that modify the behavior of the infected cell. So fungi, I know more about fungi and how they do that, but they will oftentimes produce proteins that get secreted when they're inside the plant host, for example. And those secreted proteins, which are called fungal effectors, a lot of the times those proteins are used to manipulate the host proteins that might otherwise be used as a defense against the fungus. So normally these would be host proteins that protect the plant, but if the fungus wants to stay under the radar, which biotrophic fungi do, it would say, here, I've got a protein that's going to interact with your radar protein. Why don't you just go back to sleep? What's happening here is not your concern. We're not going to cause any real damage. You just forget about us. And it's those groups of proteins that are a big focus for my lab right now. And one of the reasons that we're interested in that, not only sort of the fundamental question of, how does the fungus manipulate the host, but also it turns out that microbiome is a really good model. So that group of fungi, hundreds of fungal species, that's a good model for trying to understand emerging infectious disease. And the reason I say that is because a lot of emerging infectious disease is not due to some new species that arose from nowhere. It's due to a species of pathogen that was there all the time. Maybe it was affecting some other plant, or in the case of coronavirus, maybe it was affecting a bat or some other animal, and then it winds up in us. Or in the case of plant emerging infectious disease, maybe it was in some wildflower species, and then it jumps to a different wildflower species, or maybe it jumps to a commercially important flower species. So basically what I'm trying to say is that in studying these microbiome species, we can be trying to get a handle on predicting new emerging infectious disease. And in that situation, there are a lot less unintended consequences because we're not proposing to go in and genetically modify the fungi. We're using our understanding of this to predict what the next emerging infectious disease is going to be, and to start getting ready and prepared for that in case it turns out to be a devastating one. Again, the analogy is COVID.
Yeah, I mean, supremely relevant to right now. And it seems like also kind of an approach that would have less unintended consequences is understanding the effects of fungal effectors or, effectors of any sort and how they're inhibiting, they're kind of short-circuiting the host organism's actual immune response to defend themselves against the pathogen. That would seem like another area that would have less unintended consequences, is trying to do something about those effectors, or at least understand how they work and maybe, help the immune system, the natural immune system of the host, overcome something like that so they can't fly under the radar.
Right. And the other thing is, of course, to anticipate where a particular fungus doesn't currently infect a particular plant host, but if you look at its inventory of effectors, you can predict that it would be, maybe by not too many mutations, possible for that fungus to develop the ability to jump to a new host and potentially to thrive on that new host.
And echoes of the wisdom of Dr. Lerner with understanding this kind of evolutionary perspective of these things.
Definitely.
I'm struck by how much you can extrapolate it, not to other fungi. That's one of my big questions. Can you extrapolate these findings? But it sounds like we can extrapolate some of these basic mechanisms, kind of some of the bigger themes we've been talking about and how these pathogens work, to even other pathogens that affect other organisms like us. So really interesting to see how impactful that work is. Now, I am curious because I know you're doing this work. You also have the help of your lab and all of your students. And that's something I don't want to neglect here. So I guess tell us a little bit about the Perlin lab, how many students you're currently working with, and then if there's, a couple projects that stand out, maybe as directly related to what we're talking about, or just as supremely interesting, which I'm sure they all are to you, but just that kind of brief overview.
That's also an excellent question. And actually, I would flip it around. I would say that It's people working in my lab that make possible the work that we do. I put up slides, acknowledging the important people for the research that's been done. And I typically leave my name off. It's what do they call that? Plausible deniability. So my name's not there. But really, the reason my name's not there, typically in an acknowledgement slide, is because it's the people that are in my lab, many of whom I consider directly to be collaborators and colleagues and eventually peers, as well as my other collaborators at other institutions, that I think makes it possible to do a lot of this work. It's very hard to be a lone scientist in the modern world, I think. Really, you have to rely on the expertise and the different perspectives of a number of different people, because everybody brings to the table their experiences and their approaches to the work that we do. So I'll talk about people in my lab, and then I'll talk about actually some of the collaborators that I have that help make this work possible as well.
Terrific.
In my lab currently, I think I have about 5 or 6 PhD students. I have 1 master's student. I'm about to get another master's student in the spring. And I have, I think, 6 undergrads in the lab now. So that's kind of a big lab. It's kind of a lot of people to keep my thoughts clear on what they're doing. It's been particularly tricky since the lockdown and COVID. Basically since mid-March, we had to very carefully justify continuing to do research in the lab when they basically wanted to shut down the whole university and make everything remote. We were able to make that argument because we had ongoing government funding, and we couldn't just stop. So many different projects would have been put off. I would have had 2 PhD students that wouldn't have been able to graduate when they were supposed to. So we kept going, the grad students and I kept going until we finally started to gradually reopen labs and the university. Undergraduates were not really allowed to do that under those guidance regulations. But now everybody's back. We still do social distancing and the kinds of protective work. I mean, ironically, the kind of lab that we're in anyway, we're used to using PPE anyway, because a lot of the stuff I work with, it's good for us to protect ourselves from that, but also we like to protect our reagents, which we can consider our fungi and microbes, as reagents that we want to maintain as pure cultures and not contaminated with other kinds of things. So we're used to wearing gloves if necessary. We're used to wearing masks. We're used to disposing of things and disinfecting things. So that wasn't a big switch. Reducing the number of people in the space at a time has been a challenge, but we've been able to do it. And so far, things have worked out well, even considering the rising number of cases of COVID currently. I'm going to give a shout out to our institution. We just got a report. We've been testing at our institution, students, faculty, and staff. And at this point, I think we've had over 35,000 tests done. And we're currently at about a 2% positivity rate for COVID. So that's good. I have to say it's better than our state as a whole and better than our county, which is probably the largest county in the state. So I think we're doing a pretty good job.
I'm glad you guys have been able to adapt. And I mean, some of these findings could be directly applicable to pathogens. So we need this to continue.
In terms of projects that we're doing, I'd say the 2 or 3 main projects that we're on, we're continuing the work with those fungal effectors, trying to understand what they do in the organisms that have them normally. Kind of an offshoot of that, I've got a student who's trying to figure out whether the effectors from one fungus are specific just for that fungus, or if they might have function, if they're more universal, I guess is the question. Could they elicit more universal effects in other fungi? And so we're doing some kind of mix and match experiments in completely controlled settings, of course. We do all of our growth and infection studies in biological containment facilities, and we would never do any of those kinds of experiments outside of the lab. But I would say that we are trying to look at ways in which is a particular fungal effector from one species, does it have a more universal application that other fungi could use in their own specific host pathogen interaction, just so that we can understand, is an effector a universal product that can be used, or is it specific for the individual species? And then the third thing that we're interested in is The cellular organisms that aren't bacteria, we typically, all of those are eukaryotes. And what distinguishes them from bacteria, one of the things that distinguishes them is that they have these internal cellular structures, organelles. And one of the organelles that they, almost all of them have, are called mitochondria.
Yeah.
And mitochondria, you typically think of as sort of the powerhouse for the cell. It generates
Energy
a lot of
factories.
energy for the cell. But it's also involved in other aspects of normal cellular function. And there are a number of human diseases, for example, that involve defects in mitochondrial function. So the question is, how do mitochondria get inherited? Because they're not part of the nucleus. There's only one nucleus, typically, not always. In science, you have to be careful about making generalizations that somebody will say, oh, wait a minute, but what about this one we just found?
Right. There are always exceptions.
Always exceptions, which teach us things. That's the neat thing about science. But how do these mitochondria get inherited when the cell divides? It's a fundamental property of many organisms, including humans, that when sexual reproduction takes place, which mitochondria wind up in the offspring is controlled. Not to 100%, but a lot. So a lot of your listeners may be aware that the human egg cell is the one that decides which mitochondria wind up in the offspring. And so it's usually the mother, that's the one that makes the egg cell. It's the maternal side that usually controls which mitochondria wind up in the offspring. So even though male parents can provide some mitochondria, very few, if any, of the male mitochondria wind up being maintained in the offspring.
Is part of that just figuring out what the selection criteria is, why they would decide certain mitochondria to be passed on, the fitness of the mitochondria or something like that?
Well, see, that's an evolutionary reason why this process of control evolved.
Yeah.
Right? That's an explanation for why you might do it. It may be that certain mitochondria are more fit. Now, why should it be the maternal mitochondria that are more fit? But it may also be the idea that it's not good to have mitochondria competing with each other inside the overall big cell.
Wow.
And so it may have been the case that a system evolved whereby one of the two mating partner cells decides, okay, it's going to be my mitochondria that are the ones that survive. Any incoming mitochondria that come in, we're going to destroy them. Well, in some cases, that is what happens, right? So the egg cell, the human egg cell, actively has a way of making it hard, or if not impossible, for the male mitochondria to stick around. So it turns out that fungi, not all fungi, but many fungi also control during the mating process whose mitochondria stay and whose mitochondria go. And we think it's easier to study this idea of control of mitochondrial inheritance in a nice model system like single-celled fungi rather than a multicellular organism like a human. Easier to make the manipulations, easier maybe to put our fingers on the exact control mechanism. And so we've, with Eucelago and Sporosaurium, both of those, it turns out that that's a very interesting system that we can start to tease out the roles of certain genes and certain aspects of the mating process in controlling mitochondrial inheritance.
Yeah, it doesn't get much more fundamental than that. And it seems like a lot of your work is figuring out some of these really fundamental questions, not only in the model organisms you're working at, but then extrapolating that out to other biological organisms, including us. So really, really fascinating work. I encourage people to go read more about your work, dive into your research. There is a lot of it, and it'll certainly be an education like it was for myself, trying to keep up. It really brings out some of that rudimentary biology we all had and kind of forces you to kick it up another notch. But where can people find out more about the lab and about your work?
So if you go to, I don't have it memorized, but if you go to the University of Louisville main website, and then you go into the biology website, there is a list of faculty by name. And if you click on my name, you'll find links to all of the kinds of stuff that I do. I think it's called Mike Perlin's Universe or something like that.
Academic Universe, yeah.
Academic Universe. And so if you click on that, there's a page that talks about my research interests. It gives a kind of an overview of the things we've talked about, even a little bit more detail about the bacterial antibiotic resistance evolution stuff. And then there's a list of recent publications, and I think that's a good place for people to start. Depending upon how much scientific background a person has, and even someone that's got a pretty fundamental science background might find some of the articles that we've written a little bit, sort of, wow, what is that? I don't understand half of the words in that summary. I don't understand half the words. So rather than jumping into those, I would look at that sort of summary of the kind of research that I do that's really meant for a scientifically literate but not necessarily specialized audience, and see if any of that makes sense. And then if you want to explore other more in-depth articles, you can look at the list. And if you're not sure where to start with those, you can send me an email. I'll be happy to try and respond to your questions and at least point you in the direction to get a better overview of the kind of work that we're doing.
Yeah, I definitely felt even reading the summary that it required a couple rereads for me to really understand what I was digesting there. And as we've talked about a lot of your work, obviously going down to the gene level and examining individual proteins, it seems like to me you are very much a cautionary voice to say, hey, if we understand these mechanisms, we can't immediately assume that some, novel gene editing or some modification is going to be the best way to approach these problems. And I always appreciate voices like that because I think a lot of people, and it might be due to a lack of understanding of the exact processes, like it is admittedly for myself, I have some hesitance toward any kind of playing with genes or doing any kind of modification or editing, because I'm very worried that once you let that cat out of the bag, who knows what it's going to do to the rest of biology. And so I very much appreciate your voice that is well-informed, that seems like still saying, let's not paint with a broad brush and immediately try to meddle and change things necessarily.
Yeah, I mean, to be fair, scientists, for all the good that we've been able to accomplish and all the positive things that come out, we have a kind of a checkered track record in terms of some of the things that we've figured out and then how those were turned into technological applications that didn't always have good outcomes. And I think it's important to have a certain amount of humility. And, there I don't know if you ever saw the film Body Heat?
No, I don't think I saw that one.
It's a classic, sort of a modern, what you call a film noir. It takes place like in I think it's 1980s in Florida, but it's sort of an update of those sort of hard-boiled detective stories where there somebody gets murdered and romance, but it's dangerous romance and all that stuff. In any event, one of the characters in it is a guy whose profession is basically an arsonist, and he's talking to his lawyer. And I don't want to, spoiler alert, but the lawyer is anticipating doing something that may not be the best legal option. And he says, somebody told me that if you're out to commit a crime and there's 100 ways that something can go wrong, and you discover 50 of them, right? You anticipate 50 of them, then you're a genius. And basically the person that taught me this said, and guess what? You are no genius. And so the person that taught him that, of course, was his own attorney, the guy who's now saying, I need some advice on how to commit this crime. And the guy's telling him, look, you are not going to discover by any long shot all the things that could go wrong. So really, rethink this thing that you're trying to do. And, I think the same can be said of science. I mean, we try to anticipate everything that could possibly go wrong, but we're human beings. And sometimes the drive to move ahead on something that we think is important can blind us to the things that could go wrong. And, we really have a responsibility, all of us do, to make sure that we do the best we can to maximize the benefit and minimize the cost to society, cost to the environment, et cetera.
Yeah, like I said, I really appreciate that voice and that ethos. And I guess, do you have advice for anyone who wants to get interested in fungi or researching maybe some of the things that you're researching? Advice for maybe high school students or younger folks out there who want to get involved with this? And I know that could be a huge train of thought as well, but just any general advice for folks?
Yes, I would. From my own experience, if you're interested in fungi, I recommend that you actually get some formal training working with fungi. that's advice. What is it? Do as I say, not as I do. That's advice that I've been trying, I've been working on it, but I never had formal training working with fungi. I started working with bacteria, and I kind of fell into working with organisms that are fungi, and I've kind of, learned on the fly and try to become more sophisticated in my understanding than I was. But, I never took a formal course on fungi, ever. I would guess that we talked about this. You are somebody who likes to go out in the woods and look at mushrooms. These fungi that we work with, they're in the same general group of fungi that includes mushrooms. But I would argue that you probably have a better opportunity to identify mushrooms out in the wild and a better idea of which ones are more likely to be harmful, if you want to eat them, for example, than I would ever try to do. And part of that is that I don't have that super broad understanding of the field of mycology as much as I continue to try and learn about it and become more comfortable with it. And similarly, if you're working with fungi that infect plants, it's probably a good idea to have some formal training in working with plants too, right? If you want to look at both sides, if fungi are what interests you and what you get passionate about, and I'm pretty passionate about it, it's good to have the kind of background where you can start to make connections because you have a broader understanding of these systems that I've had to kind of work over the 30 plus years I've been doing this to gather. And part of that is that's another thing I would say to anyone that wants to go into these areas, have good people you can collaborate with. You don't have to be an expert in everything. I'm certainly not. And so talk to people that are expert in areas that you're not an expert in, because then you can gain their perspective and they can take a piece, it's kind of like division of labor. They can take a piece of the puzzle that you're not as well qualified to deal with, and you can work on the stuff that you are qualified and you can put it all together and have a much stronger understanding of what's going on. So I would say that would be advice I would give to anybody that wants to get involved with fungi or any other aspect of science.
Yeah, I think that's terrific advice, is get your fundamentals about that organism or that topic you want to learn about, and then don't be afraid to collaborate. That sounds like amazing advice for anyone who wants to get involved with this. And then I have 3 standard questions I ask all my guests, and I'm going to broaden the first one just because you're someone who works with all manner of fungi, not necessarily big showy mushrooms. But what is a mushroom or a fungi that you love or that inspires you, and why? And it doesn't necessarily mean favorite. If it's favorite, that's great. But just a mushroom or fungi that you really enjoy, and why.
Okay, so first of all, I need to make a distinction. Mushroom-forming fungi are part of a group of a larger group of fungi. Not all fungi produce mushrooms. The Bacteriology group tends to be the one that produces mushrooms. The smuts that we talked about today are part of the Bacteriology group, but they don't produce mushrooms. So if I can include fungi that are particularly fascinating, I'll talk about this group of fungi that my recent PhD graduate, William Beckerson, has moved on to work with. And it's, you may have heard of these. These are the zombie-producing fungi. So these are the fungi that infect insects. We talked about host specificity. They are exquisitely host-specific. One species of fungus will infect one particular species of insect. And what they tend to do is they tend to seriously modify the behavior of the insect. The zombie ants are the perfect example. And that's the group that my student, William, is going off to pursue with Carissa DeBecker in Florida, Central Florida, University of Central Florida. And so these are fungi, if your listeners are not familiar with, they infect the ant, and they ultimately wind up in the brain of the ant, and they modify the ant's behavior so that it winds up climbing up a stalk of grass and grasping that very tightly with its mouth parts, its mandibles. And then what happens is, as the fungus develops, eventually a mushroom-like structure does emerge from the head of the ant. And that's where the spores of the fungus get released, so that then they can infect other ants who happen to be crawling around in the same neighborhood. I think those are fascinating. I mean, I'm not aware of fungi that cause humans to behave in that manner, but they certainly can affect insects. We know that there are other organisms, not fungi, but there are organisms that can affect the way mice or rats behave around cats, make them do things that are against their best interest, like not being afraid of cats, for example. But if you think about it, all of this applies to what's the interest of the parasite or the pathogen, and how does it manipulate its host to get that end to work? So in the case of the fungus that infects ants, it makes the ant do things that will be more likely to spread the spores of the fungus to more ants. And in that other example with the mice or the rats, basically it makes the mouse not afraid of cats, so that the infected mouse gets eaten by the cat, and then the cat then can continue the life cycle, and it's repeated that way. In answer to your original question, which fungi fascinate me, that's one that I don't study that I think is particularly cool. The ones that make zombie ants.
I think that is a species of cordyceps, but
yes, you're right.
Okay, yeah. So cordyceps are, absolutely, again, mind-bending organisms, and I can tell you have a definite fascination with pathogen host interactions. And it's funny when, we start to think about what defines pathogen and host, and how are we being manipulated by all the different microorganisms that live within us? Are there already fungi that infect people and make us do things? But I don't like to think too long about that because I might not like the answers. But that's a terrific answer. The cordyceps are fascinating. And then a much bigger, broader question, but what has a relationship with fungi and fungal organisms given to you, in terms of new perspectives or new appreciations of biologic life? What has that relationship with fungi that you've developed now given to you?
Well, I try to at least instill in my students a sense that whatever you're going to do in life, it ought to be fun. Fungi have fun already in their name.
Can't go wrong.
So I think that's a key thing. Like you're going to spend your life doing something, whether for vocation or avocation, it better be fun or it's not worth doing. We have the luxury, I have to say, I feel very privileged to be paid to do something I like to do. Not everybody can say that. I wish it were true that everybody could say that. Why do a job if it's not fun? But that's a perspective that I've gotten. And I think appreciating that it's not just about humans. The perspective that the only thing that's interesting is something that directly affects humans, I think is a limited one, because we can see throughout the biological world different strategies and different directions that organisms have taken to fulfill a kind of a fundamental thing that organisms do, which is reproduce. Strip bare everything else, that's really what organisms' ultimate goal is at some level. And to be able to understand how fungi, to me, are a great model to understand sort of the strategies and ways that organisms choose to reproduce and to survive.
Absolutely. I think it does that for a lot of folks. Fungi kind of help us see through a perspective that isn't always that rather egoic hierarchical structure where humans are at the top and the most important. There are plenty of other organisms to appreciate and plenty of other ways of existing that are inherently fascinating. I love that answer as well. And then final question here, what is the lasting impact that you hope to make with your work? And that can mean, both directly the research that your lab does, but also your work in mentoring students and developing a lab like you have.
I like to be able to pay forward the kinds of mentoring and, advice and expanded mind and outlook that I received from Steve Lerner and other people that I think provided guidance as I was being trained. I want to kind of pay that forward and help students understand that there's a lot to learn out there, and there's a lot that you can do in terms of making an important impact on, if nothing else, just on human knowledge. I mean, that's part of the scientific endeavor, is to expand human knowledge, to help us understand the natural world beyond what we can just see. When I look out the window, it's more than what I see. It's also, why do I see that? Why are things happening the way that they are? And to be able to give students that I mentor the toolkit to be able to approach those questions in a systematic and, scientifically reproducible and fun way of doing things.
Well, I got to say, your students are lucky to have you. It was an absolute pleasure to get to speak with you. You were breaking down concepts to a very fundamental and approachable level. I'm really glad for the privilege of being able to speak with you, Dr. Perlin. It was a joy to have you on the show. Thank you so much for coming on the Mushroom Hour podcast.
Well, thank you, Darren. It's been a pleasure as well. And let me know if you have other questions, or people that are listening, if they want to address these things in greater detail, feel free to contact me. Appreciate it.